diagram below shows. Question 6. We've got a diagram below showing stages in the Calvin cycle of photosynthesis. It's actually a really nice, clear diagram, and if you wanted to lift this out to use as part of a diagram set when you're doing like a cue card or a mind map, you could do a lot worse. It's quite a nicely drawn one, very clear. Right, so we've got carbon dioxide coming in, joining into ribulose bisphosphate. We've got intermediates, which then goes to substance Y with the addition of ATP coming in for energy and NADPH. We get glucose out of this, and our ATP is coming back to ADP and PI on the other side to go back to ribulose bisphosphate. Okay. Name enzyme X and substance Y. So enzyme X we have up here. It is the only one that you need to know for the whole cycle set, and that is Rubisco. Because if you feel like it, you can write down ribulose bisphosphate, carboxylase, oxygenase, but I would go with Rubisco. Substance Y. We have substance Y is the one that is produced with all the stuff from stage 1, your ATP and your NADPH, and it's the one that is going to be converted into glucose, so G3P. Or you could have written glyceraldehyde 3-phosphate. Absolutely go with the full name if you're happy with it. I'm better with the 3 letters. Explain the importance of producing glucose and RUBP in stage 3. So they're saying, why do you need glucose? So you need glucose as a respiratory substrate. If you don't have that, you starve. Can't spell straight. It's closer. Oh dear, bad spelling this morning, sorry. But then you've got the ones that actually are in your setup now that you're expected to know for what glucose could be used for. So you could go for starch formation, because that's a storage set that you need to be able to do. You could say cellulose, and you could go for the very, very higher one, which is for biosynthetic pathways. Glucose just very important, obviously. And why we need to get RUBP is because it's a cycle. Okay, so RUBP feeds back into, oh dearie me, back into the cycle. Okay, and if you didn't have that, then obviously you couldn't run your cycle keeping going, so you couldn't make more glucose. Right, research has been carried out which aims to increase photosynthesis in crop plants by inserting genes for the production of prokaryote pigments into the cells. These pigments absorb wavelengths of light which are different to those absorbed by the pigments present in the crop plants. Predict what would happen to the concentrations of ATP and NADPH in the crop plant cells. Okay, so what should happen, remember ATP and NADPH are produced in the light-dependent stages. So that's the stages which involve absorption of light. So if I've added more wavelengths of light that I can absorb, well, basically this should go up and that should go up. So we've just got an increase and an increase. Okay. Moving down a little bit to get this one in. Genetically modified crops are evaluated in field trials. Certain experimental procedures are required when setting up field trials to compare GM and non-GM crops. Give one such procedure and explain how it allows valid conclusions to be drawn. Okay, so we're looking for how you would set up a field trial, and you've got a few in your list. Okay, so I would say the first one, we're looking at replication. Okay, so what we need is to ensure that there's adequate replication so that you can have a trustworthy set of results that deals with all of the differences that you have, all the variation that you have within a normal population. Okay, so replication would be essential. Okay, you then want to try and remove bias, so that's about randomization. Okay, so randomization of your plots means that you don't put them all in neat rows of here's set 1, set 2, set 3, set 4 for all the different conditions. What you would do is you'd have 1s mixed in with 2s and 3s and all of that. So we're trying to remove bias from the sample. And that might be a nice one to just use because you're just like procedure, randomization of plots, explanation, removes bias. Okay, and lastly, it would be your makeup of how you set it up. So so that's your valid kind of setup of the system. So that would be how do we put that as a simple one? Treatments. Treatment selected. Okay, so treatment selected should be of a decent range. So in this case, if it's just GM and non-GM crops, that's fine. You just need to have both of them there. But that means that it will allow you to make a comparison between the two. Okay, so replication, randomization, and your actual treatment decisions. And then give an explanation for each. Or one, obviously, for the mark. And question done.